Thank you so much for tuning in. We have a fair number of people tuning in from Zoom land, and then thanks of those who are here in person in Los Angeles, California. My name is Adam Leventhal, and I'm very excited to announce and introduce today's keynote. I believe we're calling this a summer seminar, a special summer seminar, because we usually don't do seminars over the summer. We're very honored to have Dr. Francesca Filbey. So a little bit about Dr. Filbey. She is a professor, and she also is a director of cognitive neuroscience research of addictive disorders at the Center for Brain Health, as part of the School of Behavioral and Brain Sciences at UT Dallas. She also is in University of Texas at Dallas. She also is an Adjunct Associate Professor at UT Southwestern Medical School, Department of Psychiatry, and she holds the Endowed Bert Moore Chair in Brain Health. Francesca has quite a track record of contributions to the field of addiction science, neuroscience, behavioral science, for quite some time. Just a little bit of a background about her. She got her bachelor's in arts and psychology from University of Texas at Arlington, and a master's in applied cognition and neuroscience, also there at UT Dallas. And then she got her PhD in experimental neuropsychology at the Institute of Psychiatry at King's College in London. Did a postdoc at NIMH in Bethesda. And then she has, I think for the past, let's say, 13 years, 12 years been at?
Almost 13, yeah.
Okay, 13 years has been at UT Dallas and has done a number of amazing things for their university, both with in terms of research and also in terms of leadership. She holds the Associate Provost role, which is a university-wide role there at UT Dallas to foster faculty development. And so anyways, we're really pleased to have her. Just a little bit more about her research expertise. She literally wrote the book on the neuroscience of addiction. So you should check out her book on that topic. She's been co-author of an edited book, several book chapters. She's on the editorial board of the American Journal of Drug and Alcohol Abuse. She's an active grant reviewer for a number of different funding agencies. She's a member of the board of the College on Problems of Drug Dependence, which is the main research society associated with addiction science for drugs. And she's also a member of the American College of Neuropsychopharmacology. A little bit more specifics about her goal is her research aims to advance the understanding of biobehavioral mechanisms related to addiction, especially focusing on enhancing early detection and intervention from a lifespan approach. She incorporates translational approaches from cognitive neuroscience, neuroimaging, genetics, neuropharmacology, psychology, psychiatry, and cross-cultural perspectives. And some of our current projects involve the determination of these effects using a number of different types of neuroimaging tools, even neuromodulation tools, genetics, computational modeling. And a lot of her work more recently has been on the interesting substance class of cannabis and cannabinoids. So we're really excited to have Dr. Francesca Filbey.
Thanks, Adam. I appreciate the introduction, and thank you all for coming. I've had the tremendous opportunity of meeting a lot of you these last couple of days, and I've been very impressed with the Institute of Addiction Sciences, and I feel like I'm speaking to or preaching to the choir, because the kind of work that I've always felt needed to be highlighted is the translational approach to addiction science. I think focusing on just one approach or one discipline doesn't get at the full picture. And what I hope to show you today is how we can do transdisciplinary work effectively, and that there's a lot more work to be done. And I'm so excited for you guys being here at IAS, because you are right there at the cusp of being able to do all that amazing work. So when I was asked to give today's talk, I thought about, showcasing kind of the tremendous amount of change that's happened just in the last 13 years since I started this journey in addiction science. My work has been primarily on cannabis, so the title is on cannabis. And in terms of the cannabis landscape, it's really changed so much. Just the policy surrounding cannabis, attitudes towards cannabis, but also the way that we can study the effects of cannabis on the brain. So with the neuroimaging advancements, it's really, been such a difference in just the 13 years. And so I thought, hey, why don't I just tell you guys a story of what my journey has been like and just really showcase how quickly one can really contribute to the field with collaboration, with team science. So let's begin. Ooh, let me see. Why am I not beginning? Oh, there we go. First of all of my work is funded by NIDA, the National Institute of Drug Abuse. So I'm grateful for their support all of these years. So again, this is going to be a story about my journey. Oh, sorry, that's the wrong slide there. Might be the oldest person in the room if you don't get this. This is the actual band journey. When you Google journey, as I, because I wanted to put an image in front of you of journey, they have to scroll through a million of these rock bands pictures to get at what I really try to mean, which is the journey. So when you think about a journey, at least my journey into cannabis research, I really started thinking about craving, right? So seems pretty simple. The journey begins for me with the concept of craving, but back then in the early 2000s, 2009 or so, people really didn't know what craving was all about. So why don't we think about what that means for you for a second? What is craving? Seems simple enough, right? If you can conjure up a feeling of craving, I mean, it's lunchtime, maybe it's pretty easy. Just take a look at this, right? Does that conjure up any kind of feelings, urge, strong desire? Well, that's what we call craving, right? So let's start by that feeling. So retain that feeling. Hopefully it's something like this, when you're feeling that intense desire to drink coffee, at least from that green image of the lady, right? And so craving really is that high need, high urge, strong desire to use a substance. And it's been a construct that has been shown to be clinically meaningful. So I have just two excerpts here from George Koob, who is now the director of the National Institute for Alcohol Abuse. He says about craving that it's an intense craving that follows the cue. So he's specifically talking about that high intense desire in response to exposure to some kind of stimulus that's been conditioned with that feeling of reward. And he also says that that urge is an unstoppable force that leads to drug intake and relapse. And so clinically, that's meaningful because this craving construct is what keeps people from maintaining abstinence. And then Nora Volkoff, the director of the National Institute on Drug Abuse, says that craving can reemerge even after years of abstinence. So think about that for a second. You think you have reached a level of abstinence where you're successful, but you're always at risk, right? And so how do you make sure that you achieve prolonged abstinence? That also underlies the fact that there must be some underlying neural mechanisms happening there. Even though you've been abstinent, you haven't been consuming the substance for a long time, that the changes that the consumption elicited in the brain is so long lasting that it could trigger this intense desire to use even after long-term abstinence. But when I started this journey, the term or the concept of craving was actually controversial. In fact, there's a paper from the 1970s, so this is a long ongoing controversy that said that craving concept is actually not meaningful at all, and that it doesn't really associate with behaviors, and it certainly doesn't predict relapse. And so the controversy existed for a long time. And when I started this journey into studying craving, that's what the landscape looked like. This was a review by Syed in 2000, and he really outlined why craving was so controversial. Again, so controversial when I started this journey that people wouldn't even use the term craving. They kind of skirted around the word. They would use craving in quotes, or they would say high urge, strong desire, and then craving in parentheses. So that's how it was when I started. So the things that that Syed outlined as the major contributors to why craving was so controversial at the time was because the concept is very loosely defined. How is craving, for instance, different from what we think of as desire, right? And this is kind of philosophical. They always quote philosophical excerpts when they talk about the definition of craving and how it's different from desire. But I think what is important here is that the question is this a continuum? Maybe this is just one construct, right? With craving that is maybe mild and desire being the opposite, most extreme end of the intent or intense urge to consume or take part in something. Another challenge is the scope of craving. And what exactly is the construct of craving trying to encompass? Is it the actual feeling right before you take something, or is it the right before you consume or take part in something, or is it the just the intent to use before you actually have that urge? So at what point is craving the construct that we're trying to define here? Another one is a time frame of craving. Circadian rhythms influence the amount of or the intensity of craving that people desire or people feel, and also it just varies from day to day. It also depends on the context, depends on how long they haven't they've been away from the substance or whatever the trigger is, and so that's still very much an unknown when I started this journey. And then finally, is awareness a part of craving? Can you have craving without knowing that you're craving? There was a really interesting study, and I can't remember the details now, where they looked at specific metabolites during autopsy that was that they felt was a marker that there's craving even after people pass. So that suggests there's no awareness necessary for craving. But again, these are all the reasons why craving, when I started this journey, was really controversial. So when I started, I looked at the literature and I found a lot, and I know you are not able to see that, but I hope what you're taking from this slide is the amount of behavioral studies that have shown evidence for craving for cannabis. And this is craving for cannabis in the presence of a cue or conditioned stimulus. And what's really amazing in these behavioral studies, from the review paper that I did with Sam DeWitt, showed there, is that they found craving despite the different modalities of cannabis cues. So if they showed visual cues, they did mental imagery. Despite those modalities, they found greater response to those cues through self-reported measures of craving, through galvanic skin responses, through eye tracking. And so all of these behavioral studies have really provided strong evidence that even for cannabis, that you can measure craving. And so when I saw that in the literature, I thought, well, then surely we should be able to find underlying neural mechanisms that would be associated with those responses that have been widely reported. And so while I was in a lab in Boulder, I decided to translate some of those behavioral paradigms, those cue elicited craving paradigms, into an fMRI paradigm. So while in scanner and MRI machine, we collected scans in heavy cannabis using individuals. And I want to tell you right now that all of the data that I'll be showing you are in heavy cannabis users. Heavy cannabis users, by our definition, are individuals who use cannabis daily and who've been using cannabis continuously at that rate for at least 1 year. So we're defining them as chronic, heavy using adults. So these are all individuals above the age of 18. And so we recruited those individuals, and we recruited heavy users because we thought, well, if we're going to find craving in the brain, it's going to be in people who have been using it heavily and who've been using it for a long time. We also thought, if we're going to look at craving mechanisms in the brain, we're going to want them to not be intoxicated, right? We don't want them to be acutely intoxicated. We actually want them to start withdrawing because that's going to elicit even greater craving. And the time frame for or the peak point of withdrawal symptoms for people who use cannabis is usually between day 3 and 4. And so we asked these individuals, and this is going to be for all the data that I show you today, we asked individuals to abstain for at least 3 days. Okay, so with all of that said, what you're looking at here is what individuals saw or did while they were lying there in an MRI scanner, and we're taking images of their brain. So again, we are translating the behavioral studies that were already out in the literature, and some of them were tactile cue studies. What that means is that they would expose individuals to actual cannabis paraphernalia, like the ones you see here in front of you, and they then get some kind of a response, like a subjective response, in terms of how much they are craving in the presence of those paraphernalia. So we did this exact same thing in the scanner. What you're seeing here is a pipe, which we legally collected from the police department. While the participants were in the scanner, we handed them the pipe. So the pipe was in their right hand, and the mirror above their eyes were positioned such that they actually see their own hand holding the pipe. So we were trying to make it as ecologically valid as possible. I must say that besides being a tactile cue and a visual cue, it might also have an olfactory cue because there was some residue there, and you can still smell a little bit of the marijuana. Of course, we had to control it. We had to compare it to a control object. And so we wanted a tactile control object, and we chose something that was similar in shape and color and size. So we just used a pencil for that, as shown here in this image. We did individualize the cannabis cues. So depending on their most frequently used paraphernalia is what we presented. So here's the other options that participants could choose from. We also, besides a tactile control cue, also wanted to control for any appetitive response, right? Because people are going to like something naturally. And so we also wanted to make sure that whatever we see in the brain is specific to cannabis, not just to anything that is appetitive or rewarding. Because the brain's reward system is programmed to like anything that is naturally rewarding, like food, we used fruit as our appetitive control. And we also individualized this as individuals or our participants, which fruit they preferred the most. And so they had some options there. And here are examples of what they actually saw and held while they were being scanned. Banana, apple, orange, grapes. Okay. So now for the results. If we were going to translate behavioral findings into our neuroimaging paradigm, what we would expect to find is greater response in the brain, especially in the reward areas of the brain, when the cannabis users, shown in this image here, are presented with the cannabis paraphernalia, right? And so in fact, that's what we see. You can see in this very busy image there, slices of the brain, the red areas illustrate areas of the brain that were much more active than baseline when they are presented with the cannabis pipe. And this is what it looks like in the non-using controls. So non-using controls were almost cannabis naive. They have fewer than 3 lifetime occasions of cannabis use. And so you might be asking, well, there's still some activation there. It's probably because it's a novel cue and just something that they haven't done before, which also elicits some reward response in the brain. So what I want you just to kind of take away from here is just how widespread the areas of activation is. That illustrates that a lot of different cognitive processes are involved. And then compare that to a natural reward. So our appetitive cue, such as the grapes and other fruits, as you would expect for a non-using control, this is what the reward system of the brain is programmed for, right? And so you can see that, in fact, that's what their brain responds to more so than the cannabis paraphernalia. So what would you expect in this box here? With the cannabis users, what would they look like when they're exposed to a natural reward? No activity. Well, there's a little activity, but yeah, we thought also a little less certainly than the pipe, and that's what we found. And we directly contrasted that. Okay, so here what I'm going to show you is the contrast when we looked at the pipe images versus the appetitive control. In the healthy controls, we found that the brain just did not respond more to the pipe at all in any part of the brain for cannabis compared to fruit. And so I think this is what you would expect for a cannabis user, where you would have greater activation in these areas of the brain when exposed to cannabis that's greater than what the brain was coded for, which is a natural reward. And I'll have to point out to you that these are areas of the brain that we call the prefrontal cortex, important for decision-making, reward learning, motivation. So we thought, well, okay, we're seeing all these activities in the areas of the brain that we expect, reward areas that's really driving motivation, especially when they're in the presence of a cannabis cue. But what does that really mean? What we did is then we associated that response, the brain response, to a scale questionnaire that reported marijuana-related problems. And what we found was in those prefrontal areas of the brain, like the orbitofrontal cortex, in addition to an area of the brain called the nucleus accumbens, which is important for signaling for rewards, processing reward information, that the greater the response in those two areas, the greater the marijuana problems from that scale. So it seems to be that we're tapping into something that may be meaningful in terms of how people might end up with cannabis use disorder. And I have to say that along with a collaborator, Ken Hutchison, while I was in his lab, that this pattern of activation was very familiar to me because we have seen the same when we looked at people who were heavy drinkers, such as in this slide. So you can see kind of the same areas that I was trying to point out earlier. This is, again, what we saw in cannabis users, and we saw the same in tobacco smokers also. The different image there is to reflect that this cue here was a gustatory cue, is a fancy word for taste. We actually delivered small drops of alcohol to the mouths of the individuals while they were being scanned. And in the tobacco studies, we showed them video cues, played video clips of other people using tobacco to see what the brain's response was in heavy tobacco users, and also olfactory cues, which was really fun because we had to put together this device with a lot of silicone or Teflon tubes, got their cigarettes, like asked them for one of their cigarettes. We lit up their cigarettes with, put it in a little glass container with cotton, smoked out the glass container, and then pumped air through the Teflon tube that was inserted into the participants' nostrils as they lay there in the scanner. So team effort, because that required some engineering that I did not have. So that's what led to the images that you're seeing here. And what you can see is that it doesn't matter what the modality is, right? The response is exactly the same. We also saw the same in individuals who had loss of control over eating, and as a result had high BMI, body mass index. And so these were binge eating individuals by definition. And what we did with them while they were a part of the study was presented them with taste cues of high, not just high calorie drinks, but also high in sugar and yes, just calories and sugar, and compare that to water. Again, piped small drops into their mouths. Actually, it's so small that they couldn't even really detect it. But yet their brain activation, response in the brain showed exactly what we expected, that there was greater activation in the same reward areas of the brain during the sweet, high-calorie drinks. And by the way, this is also individualized, so they just told us what they wanted, and we found it somewhere in the fridge and put it in this machine for them. It was usually some kind of soda, juice, or chocolate milk. So what was even more interesting is that when we tried to distinguish whether it was the caloric value or the sugar that they were responding to, it was actually sugar. So it was the amount of sugar that was eliciting this activation. So I thought that was really neat, that we're finding similar things in cannabis users that have been found in other substances of abuse, also now to sugar. So all of that, in terms of my journey, has been really exciting because finally, that was all pre-DSM-5. As most of you probably know, since 2013, since the transition to the DSM-5 for diagnosing substance use disorders, craving now is considered a primary symptom for the diagnosis of substance use disorder, whereas before the DSM-5, the DSM-4 did not consider it a meaningful enough clinical concept to include as a symptom. So I thought, wow, this journey is really taking me somewhere. And so I was really excited. So I know more red blobs on the screen, but I'll tell you how these red blobs are different from the previous ones. So far, I've been showing you just random activations in brain areas, right? Random activation in the front of the brain, in the back of the brain, so on and so forth. Well, as we learn more about neuroimaging of the brain and our neuroimaging techniques have advanced, we've become much more mindful in the fact that brain areas don't function in isolation. They function as brain networks. Okay, they have synchronized activity that represents the fact that they signal and process information across brain regions. So they function as networks. And so we then advanced tried to advance our understanding of the brain's response to cannabis by looking at functional connectivity of the brain, looking at these brain networks. And so what you're seeing here is work done by my postdoc, Joseph Dunlop here, where he found that in those with problems with cannabis use, so those who were by definition DSM-4 cannabis use dependent, had greater integration or connectivity between those areas of the brain than those who did not have cannabis dependence. And so it's not just that the activation was stronger, but also the integration of those regions between each other was meaningful. And we did find that the greater connection between those regions correlated with their subjective feelings of craving when we collected subjective craving. He did some really cool things. He's a statistician by training, and he wanted to look at directionality of this connectivity because he thought that they can't just be all synchronized, right? One has to first send a signal to the other and lead to these cascading events. And so what you're seeing here, and I'll walk you through it, and I'll just take you to the most important point. What he did here is what we call Granger causality analysis. And basically, like I said, it tells you the directionality of effects using lagged values of correlations between a time series of one brain area and another. And so the difference in the lag could tell you which happened first, then that then predicts the activity of the next brain area. So this image on the top shows you that yes, all of these areas depicted in the different colored circles there. All of these areas are interconnected, even though there's some directionality there. So that's all of the cannabis users all together. Here are the dependent users. So again, we were DSM-4 times here. These are the dependent users, directionality within those brain regions when they're exposed to cannabis cues. And then this is the directionality in the non-dependent users. So I'm going to highlight now kind of the wow factor here. What you're seeing here, the difference is this gap. See this empty space where there's no connectivity at all? That's between the front of the brain for decision-making in blue, and the red is the amygdala, the area of the brain that processes emotion, and the green is the insula. The insula is a part of the brain that tells you, tells you how your internal state is doing. It gives information about your internal state, interoception. So what we're seeing here is in the non-dependent users, there's no communication between those areas, prefrontal cortex, emotion part of the brain, and this internal sensory signaling part of the brain. Whereas if you have problems with cannabis use, such as in these who are dependent, there is strong connection. Those three areas of the brain are very much well integrated, signaling to each other. And maybe that's the underlying cause of the difficulties trying to refrain from use. Maybe that's what's underlying this great or continued desire to use. So we thought that that was really exciting and another way to look at this type of data. This is where we're at now in terms of all of this data, in terms of network analysis. It's very busy, but I'm just going to take you straight to the point. What you see, this is a machine learning study where we're trying to predict whether these patterns of connectivity could distinguish between cannabis users and non-cannabis users. Again, same group, same population of individuals. This is in collaboration with Xiaozi Gu from Mount Sinai. And so what she did using this technique is again looked at that pattern of connectivity using the cue-elicited craving task to differentiate cannabis users from non-users, and found that the most predictive networks that distinguish those two groups were also correlated with subjective reports of cannabis craving. But even more interesting is this graph, this image right here. What you're seeing here is the degree centrality, basically how connected these regions are in people who use cannabis from those who didn't. So these are the top 10 areas of great degree centrality. So that's graph theory network mambo jumbo for what I'm showing you here on the left. Basically, that means that these areas are influencers, right? If you look at the middle here, this is actually an example from like a social media influencer scenario where this person in the middle has many followers that they don't follow back. And that person is deemed to have a high degree of centrality. They have great influence in their social network. And so that's kind of what you can think of in terms of these areas of the brain. These are the top 10 that distinguish the non-users from those who use cannabis. And the different colors there are basically represent the different networks of the brain. And what I can tell you is that these networks are responsible for salience processing information in the outside world that is relevant to us. And also another one is a network called the default mode network. Default mode network is kind of the resting state network that kind of keeps our the rest of the brain kind of at the ready, so that you can address and monitor things that you need to respond to. Okay, so that's where we're at in terms of the network connectivity, and hopefully what I've convinced you of till now is the value of looking at these underlying mechanisms, because it can really tell us about not just what leads to the behavioral manifestations of craving, but also tells us the multidimensional processes involved in the concept of craving and why the controversy might exist. So hopefully that clarifies some of the ongoing controversy. Now, we realize also that the problem of understanding craving is that it's so highly variable. I already told you that these individuals that we study, we try to keep them very homogeneous, right? They're all heavy chronic cannabis users. They're all abstained for 3 days. They're all above the age of 18. So we try to keep them pretty similar in terms of who they are, what they do, so that we can kind of make sense of our findings. But even within a homogeneous group, there's still going to be some variability. And some of that comes from the literature where that says that the earlier you use, right, the earlier the age of onset of regular use, the greater the likelihood that you will experience these changes in the brain, that you may be more vulnerable to the effects of drugs, including cannabis. And so we looked at age of onset of regular cannabis use, and in fact, that's what we saw. We saw that the greater or the earlier onset of use, the less activation in areas of the brain for behavioral control, okay? So that's what we found there. Another one from the literature that we looked at is early life stress. So really getting at the importance of developmental period here. Early life stress was defined by their scores in using the early trauma inventory questionnaire that asks about severe traumatic experiences in childhood before the age of 18, like death in the family, hospitalization, those kinds of things, not sitting on Santa Claus's lap, although it might feel similarly traumatizing. But what we found certainly is that the activation within that region of the brain important for reward processing, nucleus accumbens, was greater with the greater number of self-reported early life trauma. And so it did seem to leave that part of the brain critical within the reward network of the brain susceptible to the effects of cannabis. Besides early life stress, we also looked at acute stress, right? So momentary stress. While they were in the scanner, we also, for lack of a better way to say this, stress them out. So we stress them out in the scanner using the an imaging version of the Trier social stress task. If you're familiar with the task, it's got a social component, it's got a cognitive component as well. So our social component was that we did the Confederate thing where we had a research assistant with a white lab coat and told the participants that it's really critical that they perform well in this task, otherwise we have to throw away their data. And also, they're continually shown how they're performing as they're doing this task. So it's really stressful, and it's cognitively stressful because it's math. Can I say any more? But they do an arithmetic equation while they're there in the scanner. This is an actual one, by the way. And they have to press the responses by button pressing, and it's timed. What you're seeing here in the blue bar is actually the amount of time that we have to respond, which is obviously going down. And this is actually controlled to the participants' ability, and we made it just a little bit harder than their actual abilities. So it was challenging, it was stressful. And so we would show them, we would give them a very stressful task, and then immediately gave that cue-elicited craving paradigm, presented them with paraphernalia to see how stress in the moment triggers cue-elicited craving. And as you would expect, it did. You see, I'm pointing out there that same region of the brain, the prefrontal cortex, specifically the orbital frontal cortex, for motivation. It was greater compared to the times when they didn't experience stress. So really showing that there is a link between stress and reward response. And that response during stress was positively correlated with the problems that the participants experienced or self-reported related to cannabis use. We also looked at the effects of genes. This was in the old days when single gene studies were still okay. But I wanted to share this with you because it was an additive effect of specific genes that we a priori hypothesized to be involved. So these have biological meaning. CNR1 is the gene that codes for the cannabinoid receptor receptors in the brain, CB1 receptors. And FAAH is fatty acid amide hydrolase gene that codes for the enzyme Fa that is important for anandamide, for removing anandamide in the brain, an endogenous cannabinoid. And what we found that the risk alleles for these two genes were correlated with response in those two areas that I already highlighted in the brain. Their response to cue-elicited during the cue-elicited craving task was greater the more of these risk alleles they have. So what you're seeing in the x-axis is the number of risk alleles, 0 meaning they have none of those. 1 would mean that they have 1 risk allele from either of these genes, and then 2 would mean that they have both risk alleles from these two genes. And on the y-axis is activation during the cue-elicited craving task. And so you'll see a positive correlation with greater risk alleles. Gender and estrogen is also important to look at. Estrogen increases dopamine synthesis, and so increases reward response. And so we compared males and females during their cue elicited craving paradigm, as well as their subjective craving. This is using principal component analysis to look at just the patterns from these large data sets. And so this on the x-axis is the brain's response to the cues. On the y-axis is subjective craving. And what you'll see is in the colored dots, I'll point it out because I'm sure it's hard to see. The pink are the female participants, males are the blue participants. And so these two components were the ones that had the greatest variance in the data that led to the greatest or explained greatest variance in the data. But it seemed like in the y-axis is when the males and females differentiated from each other significantly, which is in subjective craving. So what that tells us is that even though the neural response to cues, so you can see they're not that different in terms of the x-axis, so there's not really much difference in the neural response to cues between males and females, but on the y-axis, subjective craving, you can see that they separate more, so they're significantly different in their self-reporting. Okay, so their perception or their experience of craving is definitely greater in the females than in males. That could be interpreted in many ways. It could be that females maybe are just much more aware, more perceptive of their experiences than males. Maybe females or maybe males are underreporting some of those experiences. We don't know, and so I think that's important to delineate in future studies. On this figure to the right is the difference between just the female participants who were in their luteal stage versus the follicular stage. So the follicular stage individuals, women, are shown in the green here, and then luteal in the magenta. And similarly, on the x-axis is how they separate during neuroresponse to cues. And you can see they're almost in the same place, right? Almost overlapping. So really not much different, but their subjective cravings. So the way that they respond, their subjectively or self-report, their urge to use substances or cannabis in the presence of a cue definitely separated those females in their follicular phase versus those who were in the luteal phase. Importantly, cannabis use disorder symptoms also correlated with these two components. I know I'm running out of time, so let me know if I need to move forward. Cannabis climate is something that we're looking at currently. It's really nice that I was able to grab a lot of images that depict US versus the Netherlands because of the women's soccer happening right now. But this is in fact a study between a cross-cultural sample of cannabis users, similar types of cannabis users who are in Texas in a restrictive climate. So the climate in Texas for cannabis is not as permissive as it is in the Netherlands. And with my collaborator, Jana Kazane in University of Amsterdam, we collected the same groups of individuals. They did the exact same things in the scanner, and we just tried to see how they might differ based on the environment that they live in for cannabis. And what you're seeing here is cannabis use disorder symptom count and cannabis craving on the x-axis. But the same pattern, right? The Texas cohort is shown by the broken lines. And the Netherlands, the Dutch cohort, is in the bold lines. So what you're seeing is a strange, interesting interaction of a negative correlation in cannabis users from the Netherlands, but a positive correlation if they are from the US. So that means is that during the cue-elicited craving paradigm, it's basically what I already showed you in the US. The greater the problems with cannabis, the greater the self-reported craving, the greater the response to cues if you live in Texas. But if you live in Amsterdam, it decreases. The activation in those brain regions decrease with cannabis use disorder and also with your subjective craving. We don't know yet what to make of that, but it's an unexpected finding nonetheless. So we're still teasing that apart. We're looking at impacts of climate on cannabis attitudes, and in fact, we are seeing that. And so we feel that maybe some of this is moderating that strange interaction. Again, this is the same cohort cross-cultural sample. This is collecting information on cannabis attitudes using a questionnaire called the Cannabis Culture Questionnaire. And it asks questions based on what might be considered positive attitudes towards cannabis and negative attitudes towards cannabis from 3 levels of environment or context. They ask about country, state, friends and family, and also personal attitudes. And this is self-report. And what the take home here is that there are more positive attitudes self-reported in people, in the cannabis users in a more restricted climate. Think about that, guys. More positive when they can't access it. More negative in a more permissive climate, right? More negative when they have free access. It's been historically legal there or decriminalized since the 70s. So they don't know a different climate. So historically restrictive, historically permissive. That's not what I expected at all. But it's clear, although it's not clear in the graphs here, because the graphs are a little complicated. Last thing I want to show you is the primary motivation for use. We're also looking at medical versus recreational cannabis users, because the expectancies will probably also moderate. If they moderate behavior, they probably also moderate the underlying mechanisms. And in fact, that's what we found in terms of elicited craving. Medical or self-reported medical cannabis users. So these are the ones who are reporting primary use of cannabis to address ailments, had greater response in the brain than those who are using it recreationally. Could be comorbid with a lot of the other things they were taking cannabis for, so that's something we're also teasing apart. And we're teasing that apart using computational modeling of expectancy on craving, again, with my collaborator, Xiao Zigu. I know because of time, I'm just going to take you straight to the point. Basically, let me show you the image on B here. What it's showing you there is that the expectancy for nicotine, along with the actual delivery of nicotine, is the only thing that actually decreases craving. So expectancies matter. As you can see here, that's the bar, and it's going the negative direction because it's decreasing craving. If they are told that they are getting cannabis, but they think they are not, it doesn't change craving. If they are, have I said that correctly? If they're getting cannabis, but told that they're not, then obviously that also doesn't decrease craving. So expectancy really is important in terms of how it might affect craving. And we did some simulations to show that. And what it's showing here is basically what we found in this empirical study. Look here on the bottom right, is that this red curve here is the posterior beliefs. So this is what updates their expectancies based on what's actually happened. It actually increases when the drug is delivered, and so the discomfort is also less. And I think I'm happy to go over that in greater detail separately, but because of time, it's been an hour, I think I'm going to go ahead and move on. I'm going to skip the next 3 slides and get to kind of, this is just to show the clinical applications of elicited craving. I work with Sarah Ewing to look at psychosocial intervention, pharmacotherapy with Sherwood Brown at UT Southwestern to look at pregnenolone. We're looking at transcranial or transauricular vagus nerve stimulation with Sven Kroner. And all of that really is to show that elicited craving can be a marker for response to treatment. Across all of those 3 treatment approaches, we found that elicited response decreases with a successful response to treatment. And so the journey has taken us here. Right now, we know that there is that craving has clinical meaning. And in fact, functional magnetic fMRI cue-elicited craving paradigms has also been frequently validated as a good marker for treatment response. But the problem is the inconsistencies. We can't really utilize it or make these translations very effectively because of the inconsistencies in reporting, inconsistencies in conducting these studies. And so along with many others in the field, we put together a recommendation for how to collect this type of data and also how to report the data so that we can translate it into something that could really be more useful. All right. To my conclusions and future directions, craving could occur outside of awareness, but we need to find better ways of measuring craving besides self-report. Here are just some ideas. It's important to recognize variability. I showed some individual variability or moderators of response, but we also need to see how health factors might moderate it. Things like sleep, mood, and stress, as I already showed. And cue-elicited craving is a promising marker for response to treatment, but we also need to understand the time frame of that craving, right? Because it's important that we're capturing people all at the same time to actually make sense of how they're responding to treatment. So tracking that kind of response through EMA, ecological momentary assessments, wearable devices, and also in individuals who have been abstinent for a long time, like long-term abstinence. And then finally, cue-induced craving is a multidimensional phenomenon, so we need to understand the role of social context. Everything that I've shown you so far has been in the lab, has been while they're laying down in a scanner, cold room, very confined. That's not really ecologically valid, right? So we really need to look at social context because that's the context that people engage substance use with, especially cannabis. We have to look at how cognition feeds into this phenomenon of craving, like decision-making, inhibitory control. We have to look at expectancies, beliefs, climate and policies, and also motivation for use. With that, I think that you guys are all well poised to do all the things I just listed. Don't stop believing. That's the journey hasn't ended yet. I think with the clinical research core, you can really stimulate a lot of these studies, activate the people who are interested in doing these studies by providing the necessary support, training, and access to the instrumentation. Everything I've done is not done by me alone. And I tried to showcase all of my investigators, but that's not even all of them. So it's important to really have the expertise in the different areas. I am not trained as a computational modeling person, so I gloss over the computational modeling part because my computational modeling collaborator is the one who did that work and just shows you how much more we can learn if we engage people from different areas. So it's team science, and definitely one that NIH recognizes. And this is just the different fields that NIDA would be interested in terms of funding a P grant. And then, of course, we have to make sure the team science, especially in addiction sciences, continues, because it's the only way we could chip at the addiction stigma, right? Is if we continue to engage people who think that it's important to conduct the research that we do. And so training the next generation through the various mechanisms that I've listed here is just an example maybe of how to do that. With that said, I'm sorry for taking so long, but thank you for your attention and your patience. These are all the people who do the work in my lab, so special thanks to them. Thanks, guys.